Hello dear aspirants, welcome to GDC online classes. In this session, we are going to know about another new important and interesting topic, that is investigation of biogenetic studies. Investigation of biogenetic studies. Yes, I think you have covered up to the last class that what we have discussed in the up to previous classes. I think if you are up to date or if you have completed all of my videos, so you got to know that the last class, that is in biogenetic pathways, we have read about biogenetic pathways or biogenetic studies. So in that we have seen from the cellular level to higher plant metabolic pathways. We have seen so many metabolic pathways with respect to the cellular level, coming from the Calvin cycle, Krebs cycle, pentose phosphate cycle, beta oxidation, glycolysis, gluconeogenesis. And after that we have seen some of the higher metabolic pathways like shikimic acid pathway, acetate malonate pathway. Some of the important pathways we have got, which are very important for the plant survival, growth and development, as well as to get a medicine from the plant itself. We have gone through with the secondary metabolite pathways. Yes, but coming to this session, we are going to know about the investigation of such pathways, investigation of such biogenetic studies. Yes, yes, let's start the session. Yes, first of all, let us know what is the need of investigation of these biogenetic studies. We know that the biogenetic studies are nothing but the pathways which are lead to formation of some end products called metabolites. Some end products called metabolites. So maybe they are primary or secondary metabolites. The primary metabolites are very important for the plant physiological function. But whereas these primary metabolites are the extension for the secondary metabolitic pathway, or the primary metabolites are the precursor or starting products for the secondary metabolitic pathway. So we have learned all these things from the glucose, from the photosynthesis to formation of alkaloids, glycosides and some other secondary metabolites we have seen in the last class, right my dear students? Yes, but what is the need of investigating these biogenetic studies? So I'll ask you a question. When any case or when any scene or when anything, in which conditions it need to be investigated, it need to be examined? No, but in which condition the investigation has to be done, in which condition a thing should be examined? Yes, so that you got the answer, I think so. Yes, in these biogenetic studies are very important cycles or pathways in order to get the primary and secondary metabolites, because these secondary metabolites are nothing but alkaloids, glycosides, flavonoids, resins. So from these alkaloids, resins and all these things, we are preparing dose forms. So in order to get these secondary metabolites, we should examine or investigate the biogenetic studies with respect to the each and every step, in order to know and to make changes and to examine how the pathway is carried out. Clear? Yes, nothing but the investigation of biogenetic studies need to be done in order to understand how the pathways are carried out and what type of intermediates are involving and what end products are going to be formed. How can you say, and what are the end products of shikimic acid? What is the end product of glycolysis? What is the end product of gluconeogenesis? Why only the alcohol? Why only phenylalanine, tyrosine, and tryptophan formed in shikimic acid pathway? Why the fatty acids are formed in malonate pathway? Why isoprenoids are formed in malonate pathway? How can you know that? Because of some of the people or some of the scientists has investigated with help of some techniques, like we are going to discuss. Okay, so with the help of some techniques like radioactive radio tracer techniques, radioactive isotope techniques, So by such techniques, they had investigated all the pathways to know each and every step and the end product and the precursor and what enzyme is involved. So by such efforts, now we are studying different types of pathways. So you got to know that, because after investigating, after knowing all these things only, we got to know the cycles. Who said the glycolysis has this end product, the gluconeogenesis has this end product, and these type of pathway are having this end products? Because after investigation only, let us see what are the methods of investigation and what does meant by the investigation. Yes, I think you got to know that, because of these biogenetic studies only, or investigation of by this biogenetic studies only, we got to know about all the pathways. Is that clear, my dear students? Yes, first you should understand what is the need and why it should be there for to investigate the biogenetic studies, or why the investigation is for the biogenetic studies. Yes, I think you had that particular foundation regarding the why and what and how the investigation of biogenetic studies. Let us see that. Yes, we all know that living plants are considered as a biosynthetic laboratories. Why they are considered biosynthetic laboratories? Because themselves they produce, because they are living organisms, and they are able to produce their own primary and secondary metabolites. For that reason, they themselves they are not dependent, they are independent, but they are some point of time they depend on external factors. They are not dependent on other living organisms. They can prepare their own wood. For that reason, they are biosynthetic laboratories, because they produce primary and secondary metabolites for their need. And all organisms need to transform and interconvert vast number of organic compounds to enable them to live, grow and reproduce. Yes, that's what I said. In the biosynthetic pathways, either a catabolism or anabolism should be there, either formation of a new organic compounds or breakdown of that newly formed organic compound to get energy. So this process will be carried throughout the life cycle in order to reproduce, live and to sustain and develop and growth. Is it clear, my dear students? Yes. So that in order to do all these things, to enable them to live and grow and reproduce, they should have a set of reactions or set of steps are included, or set of steps are called as pathways, biosynthetic pathways. Yes, they need to provide themselves with energy in the form of ATP. Yes, we have seen that the ADP, NADP has liberated in so many of the cellular level pathways, because they need to grow, plant need to live, plant need to reproduce. Okay, and a supply of building blocks to construct their own tissues. After getting energy, after energy is liberated from the ATP or stored in the form of ATP, so they build the block, they have some blocks to build their own structures. So this is a basic protocol. So this is a basic phenomenon how it is going to be done. Yes, an integrated network of enzyme mediated and carefully regulated chemical reaction is used for this purpose. Yes, nothing but how. So in other terms, we're explaining what does it, or what does mean by and biosynthetic pathways. Yes, in the biosynthetic pathways, in every pathway there is an enzyme, so which is enhanced, or which will change the rate of reaction. Okay, so yes, obviously every pathway is enzyme mediated, and also it is a regulated chemical reaction. Yes, and used for the purpose collectively known as intermediary metabolism, and the pathways involved is called as metabolic pathways. Simply, up to now I have explained about, or I have described what you meant by the metabolic pathways or metabolism. Clear? Yes, and there are various types, like, yes, we know that cyclic pathways, state pathways, glycolysis, Krebs cycle and other pathways. All these are having such type of protocol only, such type of reactions only, such type of enzyme mediated reactions only. Clear? Yes. So this is all about the biosynthetic pathways, because in order to live and reproduce and to grow and develop, they have, or then they must and should definitely they should have these pathways or metabolic pathways. But how can you say that this pathway is perfect? From this pathway only these compounds are synthesized. How can you say that? From this pathway, these are the intermediates. From this pathway, these are the key intermediates. Some from this pathway, these are precursors. How can you know that? Even though I am telling, even though some of the senior scientists were telling that, so this is a starting precursors. Even they also, how they know about these things? Because of they have investigated these pathways by means of some techniques like all these. Is that clear? Yes, simply in one word I'll say that yes, in order to know or in order to examine each and everything which is involved in the biogenetic pathways, we require some of the techniques for the investigation of biogenetic studies. Yes, yes, we'll see. And what are the techniques or what are the methods which are involved in the investigation of biogenetic studies? Yes, we all know that use of isolated organ, that's the one method. Grafting method, use of mutant strain, enzymatic method, tracer techniques. So these are the 5 techniques. Apart from that, we are having some other techniques, but these are most prominent and significant techniques for to investigate the biogenetic studies. Clear? We'll see each one in detail so that you can understand so which method it is and what is the application of it in it. Clear? Yes, but by the end of this slide you got to know that what is the need of biogenetic investigation of biogenetic studies. Yes, immediately should your mind should strive for, yes, the biogenetic studies should be investigated in order to know the what is a precursor, what is a key intermediate, and what is the end product, which enzyme is involving, which enzyme is changing the rate of reaction. So in order to know all such type of factors or all such type of things, we should have a methods for to investigate the biogenetic studies. Is that clear, my students? Yes, and also some techniques like some techniques like use of isolated organ grafting method. By that also we are not only knowing about the all these things like precursor, end product, intermediates, and also we can also assess or we can also confirm the site of synthesis. Is that clear? Yes, we'll see all together, with all our enthusiasm, with all our interest towards cracking the GPAT. Clear? Yes, you're going to success definitely if you listen, if you catch the concept. Clear? Yes, we'll see the first one, that is use of isolated organ or tissue or cells. Yes, the cultivation of isolated. Yes, so the name itself saying that we are investigating in this. We are having 5 methods to investigate biogenetic studies. In this first method, we are investigating the biogenetic studies by utilizing the isolated organ or tissue or a cell. We can take any one of them. But what we should do, yes, cultivation of isolated organ and tissues of plants eliminates the interference from the other parts of the plant which may produce secondary changes in the metabolites. Is that clear? Yes. If at all isolation, if the particular plant or part or tissue or cell, if it is still attaches or if it is still into that plant, there will be a proper metabolism pathway or metabolic pathway will be carried. But if you removed or isolated, the pathway or the end products will be changed. Yes, so that by isolating such type of plant parts, we are going to know something. Let us see. It can be used for feeding experiments in conjunction with label compounds, and also it is used for determination of site of synthesis of particular compounds. Yes, I'll tell you an example. Yes, if you are by taking this statement into consideration, use of isolated organ. Yes, I am taking a isolated part. I am taking an isolated part. If after further cultivation, after further storage of this particular organ or tissue or cell, and after further treatment in order to develop a shoot or any of the secondary metabolites, even though I'll tell you that yes, I'm taking a part, I'm taking an isolated organ, after several protocols to make it into a specific thing to get or to obtain a secondary metabolites. Simple, I'll tell you that if I'm taking a leaf here, if I after further process, after cultivation, after preserving, after providing some nutrient media. Okay, I'm taking isolated tissue, or simply I have taken a leaf. Okay, if I'm taking leaf, and after that, in order to make it into survive, I provide some of the nutrients, water, and all these things. Yes, if after the cultivation, after the storage, or after the some process, if the particular plant, or if the particular part is having, or if the particular plant is having some secondary metabolites, yes, I have taken leaf, I have cultivated in some in vitro cultivation I have done. Okay, and after that cultivation, if I tested these leaf for confirmation of some secondary metabolites, yes, think that it is an alkaloid leaf. Okay, I don't know that, I don't know whether the leaf consisting of the alkaloids or not, but after this leaf, further process, after collection, after isolating of this particular leaf, okay, I don't know that whether it is having a secondary metabolites or not, but after isolating this leaf, after incorporating a labeled compound, then after it should be cultivated or should be grown. If after the further process to that leaf, after several days, if it is produced or if gives a positive test, a positive results for this test, if suppose I performed alkaloid test, this is an alkaloid plant, but I don't know whether the leaf is having an alkaloids or not, but after isolating, after putting into favorable conditions for several days, if I tested for alkaloidal confirmation, if the particular leaf gives positive results, which means or which indicates the site of synthesis, in simple word, that's what I'm trying to prove is here. Yes, I think it is a little bit confused, but I'll directly say that, if I have taken as an isolated organ, if it gives a positive results for a specific secondary metabolitic test, if I'm taken an alkaloid plant leaf, if it gives a positive results for alkaloid test, so that we can confirm the site of synthesis in the leaf itself. If we have taken another part of the plant, if you have taken any of the root itself, after testing to the root for the specific secondary metabolite test like alkaloid glycoside test, if it gives a positive results, so that the site of synthesis will be in the roots. So like how we, by this method, by this use of isolated organ method, we can determine the site of synthesis in which part of the plant the second metabolite is synthesized, because we are taking any other part of the plant, then we are tested for the confirmation of the secondary metabolites. So if at all it gives a positive results, so that can be confirmed for the site of synthesis. Clear? Yes. Yeah, isolated shoots of plants when placed in a suitable solution of water. Yes, the protocol. Okay, so we have taken isolated shoots of a plant, we have taken a shoot. Okay, then it should be placed in a water or a suitable solution, depends upon the nature of the plant, will usually remain target. Yes, it usually remain target for some days. It won't have any of the other metabolism or any other further process some days, and during this time presumably have a normal metabolism. Yes, you have provided the water and other required solution, it has a normal metabolism just to survive. Okay, soon however a pathological metabolism commences. When the metabolism commences, the techniques can be refined by aseptically connecting to the end of shoot of reservoir. Yes, first when we are placing the plant into a normal water solution, should have a normal metabolism, but when the nutrients are degraded, when nutrients are reduced, the plant condition gets into pathological conditions. Yes, because it is degraded, all the nutrients are getting out from the plant or shoot. So in such cases, so this shoot is transferred aseptically into nutrient media. Okay, if sterile nutrient media, when the shoots will remain normal for much longer period, then after such shoot often develop root at the cut ends. Yes, simply we have taken a shoot, we just put it into water. In such, at that time it has a normal metabolism. When the nutrient inside the plant gets exhausted, then we should transfer into nutrient media in order to avoid from the pathological metabolism. if the plant has the ability to regenerate the root itself, so that we can confirm, so at the root itself, at the shoot itself, the metabolism takes place, the secondary metabolites get synthesized. So this is how we confirm. Simply, I'll tell you that the technique has been recently used for study of biogenesis of hyperforin in hypericum perforatum. Yes, isolated leaves can be similarly maintained. Yes, exactly like this procedure. Okay, after doing same protocol to the hypericum perforatum plant, we have confirmed that hyperforin was synthesized in leaves, because here itself site of synthesis can be known. Okay, this is how by this method we can attain. Okay, we can know the site of synthesis. And the best example that petal disc have been used for investigation of biosynthesis of oil of rose, because we are getting the rose oil from the rose petals only, because we know that the site of synthesis will be in rose petals. Okay, so in order to prove this statement, prove this method, we are taking a best example of rose oil. Clear? Yes, simply you should know that. So by the use of isolated organ, we do that some procedure. So after that procedure, we got to know the determination of site of synthesis. Clear? So this is what we need to know, or what we need to learn from the method, from this method. Okay. Yes. We'll see the next method. Yes, grafting method. Yes, in the first method, we should we got to know that site of synthesis, where the synthesis has started, basic synthesis. But coming to this grafting method, we all know that grafting nothing but a joining or joining of two different species. But before that, you should know that the one which is connected on the upper portion, that is called as scission. Okay, and whereas we used to place, or which is attached to the ground or surface or soil, that is known as stalk. Okay, where we place the support one is stalk, the upper one is a scission. Yes, yes. So what we know, or what we go how can we know that the grafting method will investigate the biogenetic studies? We will see. Grafting techniques have a considerable use of biosynthetic studies. Yes, particularly for the determination of site of both primary and secondary metabolism of some secondary plant products. Yes, in that we have seen, in the first one we have seen that site of synthesis. But here we can know the secondary metabolites, exactly we can know. Okay, in the first method we can know that in which part the usual synthesis is going to be done. But here itself, in this grafting method, we know that the secondary metabolics, in which part the secondary metabolite is going to be formed. Yes, in order to confirm that, we will take an example. Alkaloid formation by the grafting plants has been extensively studied. Yes, to support this statement, to support this method, we have a best example of alkaloid plants like nicotine. Okay, nicotiana, that is the tropane alkaloid producing Solanaceae. So we have studies, we have applied this grafting method in one of the plant called nicotiana, belongs to the family of Solanaceae. Yes, thus the tomato station has grafted on the Datura stalk accumulate tropane alkaloids, whereas the Datura sessions on the tomato stalk contains small amounts of tropane alkaloid. Yes, in this we have taken example of two plants. Okay, for to prove this method, the Datura and tomato. By taking tomato season on the Datura stalk, if I take the Datura here and the tomato plant right over here on the upper part, the yield of the Datura or tropane alkaloids were increased. But if I change, or vice versa, if I change the position or the nature of the both plants, if I use the Datura as a season and tomato as a stalk, the amount will be reduced. So this is what you got to know from this grafting method. Simply in the grafting method, so we can know the site of secondary metabolic synthesis. Okay, yeah, so that if I for this statement I have taken two examples, tomato season, the Datura stalk. If I use the tomato as a season, okay, so the Datura as a stalk, the tropane alkaloids, the alkaloid content will be increased. If at all, if I reverse this position, then the amount will be reduced. So by that you got to know, the synthesis concentration also can be reduced. Yes, so secondary metabolite site and also concentration of the secondary metabolites can also be varied with respect to the this method. Okay. However, interspecific graphs, yes, interspecific graphs involving the 𝘂𝙬𝑟𝑜𝑥 and the 𝘂𝙬𝑟𝑜 demonstrate the secondary modification of alkaloids do occur in aerial parts of this plant. Yes, if you interspecific graphs, like within the same species, datura same genus compounds or same species like Datura ferox and Datura salmonum, both are having same genus. Okay, but even though the same species has a grafting method, if we subject same species to the grafting method also, we can have different changes, or the site of synthesis can also be varied, after subjecting. Even though we can see this grafting method effect on interspecific graphs like Datura ferox and Datura salmonum, if you do the grafting to these species, okay, the synthesis will be in the aerial parts of these plants. Okay, so this has been demonstrated conclusively by feeding the alkaloidal free session of D-ferox on Cypomandra betaceae stocks with hyoscine. On subsequent analysis, hyoscine was isolated from the leaves. Yes, so nothing but so in order to fetch this statement, so Datura ferox and Datura salmonum were both are same species. Okay, even though if we change the season and stock, the pattern will be, or the site of synthesis will be in aerial part. Okay, but in order to confirm this statement, we have taken another experiment, that is, if the Datura ferox is again grafted on other plants like Cypomandra betaceae, okay, the site of synthesis will be in the leaves, so the pattern will be changed. So by this grafting method, we can investigate the site of synthesis and concentration of the secondary metabolites. Clear? Yes. So that's about the grafting methods. Next one is use of mutant strain. Okay, so up to now we have covered two things. First one is use of isolate organ. Next one is grafting method, and third one is use of mutant strain. How can we assess, or how can we investigate by using mutant strain? Yes, we'll see. Mutant strain play a crucial role in biogenetic studies in plants by providing insights to the gene function. Yes, the mutant strain will give an insights regarding the gene function, so that if you know the gene function, you can alter whatever you want, so that you can change the end product of the particular metabolism, or you can change the intermediate, or you can change whatever, because gene function, you are getting insights for gene function. Okay, and the genetic pathways, how these are done, and they triad inheritance, nothing but similar things. Triad means similar compounds or similar species. Okay, triad inheritance, so that we got to know all these by means of mutant strain, use of by mutant strains. Okay, this mutant strains of lower plants, like what are the types of mutant strains that we are using? Fungi, microorganisms are produced in nature with a normal metabolic pathways are gently affected. Yes, to know all the insights of gene function and genetic pathways, we use a fungi or microorganisms as the mutant strains, which can affect the pathway very gently. Clear? Yes, such mutant strain metabolites are found in intermediate stage and need artificial supply of other intermediate. Yes, as this type of fungi or microorganisms are found in intermediate stage of the pathway, so in order to fetch or in order to support these things, artificial supply of another intermediate get be incorporated, so that the entire mutant strain get be carried throughout the process, so that we can examine the genetic pathways and the pattern, everything gets examined. Okay, such mutant strain can be introduced in the biosynthetic studies of natural products. Clear? Simply nothing but in the plant life cycle, in the plant, in the earliest stages, we used to incorporate a fungi or microorganisms as a label compound, then we can examine for throughout the life of the plant. Okay, yes. So what are the things we can know by means of the mutant strain method? Okay, so we can know, or we can illustrate the pathway. So by means of the pathway, yes, so pathway can be illustrated, or pathway illustration can be done. Gene function analysis, how these genes, or yes, that's what we meant, how the gene function. We can assess the gene function genetic pathway so that we can analyze the gene function. Stress response studies under stress. What are the changes? What is the rate of synthesis or what is the rate of metabolism? All these things can be analyzed or assessed by means of the use of mutant strains. Okay, and also trait mapping and quantitative genetics. Trait mapping and quantitative genetics. We can assess the all the genetics or all the factors or all the by means of quantitative. Okay, in the all the genes we can count or we can quantitatively we can assess the genetics in the plant. Clear? Yes, this is about the use of mutant strains. Yes, we'll see the enzymatic method. In the enzymatic method, there's a fourth method that we use to investigate the biogenetic studies. It's simply, in the name just saying that we are subjecting the enzymes to know or to investigate the biogenetic studies. Yes, enzymatic methods play a crucial role in biogenetic studies, offering a powerful tools for manipulating, detecting, characterizing the biological molecules and process with high precision specificity. Exactly, because nothing but the biological molecules are nothing but the enzymes or other intermediates. So this enzymatic method becomes a powerful tool to manipulate and detect the characterizing the biological molecules and process with high precision and specificity. And the enzymatic methods in plant biology studies are crucial for to understand the aspects like plant biology, including growth, development, metabolism, and stress responses. All these things can be assessed by means of enzymatic method. Yes, and by this enzymatic method, so nothing but we used to focus on some enzymes. If you observed or not, in the glycolysis and gluconeogenesis pathway, only change is that some of the enzymes which will change the entire reactions, which will change the entire end product. So this is how we can also take the enzymes as a base for to investigate the biogenetic studies. Yes, enzyme inhibition studies. So by this method we can know the enzyme inhibition studies to investigate the effect of inhibitors on plant enzymes. Yes, how some inhibitors, even though it's a plant growth inhibitors or some other inhibitors, shows effect on plant enzyme that involved in the various metabolic pathways and signaling cascades. These studies help illustrate the enzyme function, regulatory mechanisms, and potential targets and herbicides and pesticides and pharmaceuticals. Yes, in this, so because we are subjecting the enzyme, we are taking a enzyme as a base, so that if you focus on enzyme, okay, especially we can know enzyme inhibition studies, which thing is inhibiting. So after inhibition, what are the changes the plant the pathway is goes. Okay. Yes, the metabolic enzyme profiling. Metabolic enzyme profiling. The metabolic enzyme profiling involves the metabolic enzyme profiling involves quantifying the activities of enzyme. So which enzyme producing which amount of action, how much energy it is producing, it leads to formation how much of energy, how many ATPs. Okay, in plant metabolic pathways to understand metabolic regulation, fluxes, and responses to genetic environmental perturbations. Yes, so these assess provide a valuable information for metabolic engineering, crop improvement, biotechnological applications in agriculture. Okay, simple. We can know enzyme inhibition studies, and also we can quantify the activities of an enzyme and other we can see. And last, enzyme activities assay. Enzyme activity assays. Yes, it measures the activities of various enzymes which are involved in plant metabolism. Enzymes in primary metabolic pathways like glycolysis, food. Yes, that's what I said. Simply, wherever the enzyme involves, all the studies can be carried out. That is nothing but enzymatic method. So whether it's an enzymatic inhibition studies, enzyme profiling, and enzyme activity assays. Clear? Yes. So that by this activity assays, phenyl propionate pathway can be assessed, terpenoid pathway. All these give a very good insights to understand the different involvement or the importance of enzymes in the investigation of biogenetic pathways. Is that clear, my dear students? Yes, I hope you understand these methods to investigate the biogenetic studies. As this is for today's session. In the upcoming session, we'll discuss one of the very important technique and the vast one, that is radioactive isotopes, in the upcoming session. Yes, students, thank you. Have a great day.